questions? I don't know, might be online. Okay, well, maybe we can move on now, and the next talk, which is going to be given inside, is Frédéric Thevenet. As he mentioned in the beginning, he's professor at the IMT North Europe Institute, and he's going to talk about. So once again, the next talk is going to be given on site by Frédéric Thevenet. Frédéric is a full professor at the Institute IMT North Europe, and he's going to talk about the interaction of Gobi dust and acetic acid, and update the information about the uptake and the mechanism under atmospheric conditions.
Thank you, Menales, for this introduction. So, the results I will present today are extracted from the very nice PhD work carried out by Shenje Wang under the supervision of Manolis Romanias and I for IMT Europe, and Antoine Rousseau from Laboratory of Plasma Physics in École Polytechnique. So, looking at the interaction of acetic acid with Gobi dust is a way to explore how a surface of natural dust can be impacted, coated, or maybe aged by the presence of organic on its surface. So let's start with the obvious. We all know that the dust composition distributes with a great variety, and natural mineral dust are mixtures of various mineral phases. If we pay attention to silica, we can see that, for instance, in the Northern America, it's something like 3/4 of silica. When looking at dust from Sahara Desert or Arabic Desert, it may exceed 90%, but in the case of Chinese dust, silica content is approximately 1/3, which is lower than the others. And how far is it interesting to have such a low silica amount in a mineral dust? Maybe it can lead to a higher ability for uptake of gases, or a higher ability also for being aged or impacted by organics. How far? Because silica is pointed as the less reactive mineral by various authors regarding the uptake, the absorption, and maybe also the reaction. And another driver to understand the behavior of mineral dust is the aluminum over silicon ratio. And it has been shown by Zinedine on a quite significant diversity of dust that the highest this ratio, the best the uptake toward different gases. So if we look at it, Gobi has a higher ratio for aluminum over silicon compared to other kind of well-known Northern American or Saharan or Arabic dust. So it may be a good candidate to be aged. Now, if we look on the VOC side, why choosing acetic acid? Because it's a good candidate for being taken up on dust. If we compare the ability of acetic acid to be taken up with compared to other light oxygenated VOCs, such as methanol and formaldehyde, there are some works that point at the fact that the initial uptake coefficient of acetic acid is larger by 1 order of magnitude compared to other light oxygenated VOCs. And it is generally reported with a higher uptake coefficient, but also with a stronger interaction with the surface of the Gobi dust. So it means that acetic acid can possibly impact the surface of Gobi dust, and it's a good way of investigating how a dust surface can be coated by organics. It may occupy surface reactive site, maybe for long term, we don't know, maybe permanent term. And anyway, it's worth looking at how it may change the surface properties of dust. So if we come back to the dust we have worked with, Gobi dust, the particle size, hello Sonia Castillo, the particle size is lower than 100 micron. There are natural samples that have been sieved, and the surface area is 10.5 meters square per gram, which typically compares with other kind of mineral dust or synthetic metal oxides or carbonates. The composition has been characterized by X-ray diffraction and X-ray fluorescence for phases and elemental composition, and we can point at the quite low presence comparatively to other dust of silica, a very interesting presence of calcium carbonate, and the presence also of titanium dioxide, but that would not be as affirmative as my former colleagues about the photoactivity of titanium dioxide in mineral dust. And this is going to be discussed in a bit more details by Jérôme Lannes, discussing about the photo-enhanced uptake of SO2 on volcanic dust. And there is a bunch of diverse aluminosilicates that are responsible for the high aluminum over silicon ratio of this dust. So how it looks like? It's quite irregular in shape. Most of them are bulky, looking like coconuts. Some other are flat, like flakes, and they are generally contrasted in size, ranging from 1 micron to tens of microns. So, what's the experimental setups we used to characterize this interaction? We have first to prepare our gas flow. I mean, we've got carrier gas, namely synthetic air that flows through water line and acetic acid line. Both are initially at the liquid form in impingers, regulated temperatures, and then we can get the targeted amount of moisture, targeted amount of targeted concentration of acetic acid. 2 reactors. One is a gas-phase kinetic measurement reactor. It's a U-shaped reactor here, where the dust sample is set, a few milligrams, and it's connected to a bypass, and both can flow through a gas cell of Fourier transform infrared spectrometer to characterize downstream what exits from the reactor. It means that we guess what happened inside the reactor using this setup. The other setup is a look inside the reactor. It's a drift cell for diffuse reflectance infrared Fourier transform spectrometry. The sample is set here. It is exposed to the gas phase, and the infrared beam hits the surface of the sample, get absorbed by some organics present on the coatings, maybe, and reflected, and we analyze the reflected signal. So it addresses the adsorbed phase. So now that we have a protocol, we have a setup, we need a protocol to operate the setup and make a focus on the protocol we have used with the U-shaped reactor. It echoes the protocol that surprisingly my knowledge has shown a few minutes ago. This protocol is based on the temporal profiles of acetic acid concentration as a function of time. Step 0 is the thermal pretreatment. It's not depicted here. It's just to remove some species that could be present on the surface. Step 1 is stabilizing the concentration of acetic acid in the bypass, and then suddenly the flow is sent through the U-shaped reactor, and we've got, as seen earlier, an uptake. If we wait till the end of the uptake process of acetic acid onto the Corby dust, it's supposed to return, if not reactive, if not steady state uptake, to the initial concentration. That's what happens for this kind of dust here. Then we check again the initial concentration to make sure nothing has changed, and we shut down the concentration of acetic acid, and we flow air to sweep the sample that had been contaminated by acetic acid, and to check whether we can get back some acetic acid that would consist in the reversible fractions, I mean weakly interacting molecules that could leave the surface while the concentration is decreased. So, doesn't work anymore. Yes. So what can we do with this kind of profile? We can go for an integration of that peak that leads us to the total number of acetic acid molecules stored on the surface during adsorption. We can go for another integration of that peak for the reversible fraction, and we can see that the reversible fraction is much lower than the total, which means that, well, generally reversible fraction is related to molecularly adsorbed acetic acid. We will see whether it's the case or not. If we make the difference between both fractions, it means that we can access and quantify the irreversible fraction, the fraction that remains on the surface while you have shut down the concentration of acetic acid. So the first conclusion we can make here is that some acetic acid remain on the surface, but how long, how intensively? The first question we can also wonder here is how, since we have 2 populations of acetic acid molecule on the surface, how do they populate? Do they populate with the same rate, with the same way, or is it a sequential population? To answer that question, we can go for a partial uptake. I mean, you recognize here the uptake peak, but we can make a decision of shutting down the acetic acid flow before the equilibrium is reached. So it means we have not reached the equilibrium, we have not completely covered the surface. In that case, we have covered 67% of what has been covered while waiting the equilibrium. And we can have a look to the 2 populations of acetic acid on the surface. Interestingly, the irreversible fraction is already completed up to 93%, which means that they are covered primarily, while the reversible fraction is only completed up to 25%. So it means that the irreversibly taken fraction reaches equilibrium faster, and it says that the kinetic for populating the surface sites corresponding to irreversible fraction are covered first, or a bit before the reversible fraction. Seeing that result, we would have the idea of saying, well, one corresponds to this option, the other one corresponds to chemist option. Maybe it's not that simple, and we'll have a look. To address that question, we have determined the adsorption isotherm of both fractions of acetic acid on the surface. So this is the number of molecules of acetic acid as a function of acetic acid concentration. Black for the irreversible fraction, black, both are black. Circles for irreversible fraction and triangles for the reversible one. The reversible fraction shows a quite typical Langmuir fit and suggests that it may be a physisorptive process, confirming that it may be molecularly adsorbed species consisting in the reversible fraction. But looking at the irreversible fraction, clearly it's not a Langmuir shape for two reasons. First, it poorly varies with the concentration. And second, we already have a quite significant coverage of the surface by irreversible fraction for the very lowest gas concentration. So it means that, okay, the reversible adsorb fraction is molecularly physisorbed acetic acid. We can almost be sure of that. But the irreversibly taken fraction can be either a chemisorbed one or a strongly physisorbed fraction. To answer that question, we have to move to drift and looking at what happens on the surface. So this graph displays the typical infrared spectrum of acetic acid for different adsorption time on Gobi dust. We can see on the left side broad bands for OH stretching, always bothering the infrared spectrum. On the extreme left side, negative bands corresponding to the removal of hydroxide groups. And we've got different bands that can be assigned to molecularly physisorbed acetic acid, either as monomer interacting with the OH group, or sometimes as dimer. You can also have an interaction of a second molecule with a first one adsorbed. Okay, if now we go one step further, we can also see some other bands with blue arrows here that correspond to surface acetates, which corresponding to a bidentate structure, acetates coordinated on the surface. And this mode is very interesting. If now we go for flushing, this is the beeping curve now, we can see that while flushing, we have a decrease of monomer and dimer. So but not a complete decrease. I mean, they still remain on the surface. So this adsorption mode, monomer and dimer, probably contribute to the reversible fraction, but also to part of the irreversible fraction. Anyway, while flushing, the surface acetates are not affected at all. So clearly they contribute to the irreversible fraction. If we make the system a bit more complex, we can introduce 20% of RH. So why 20%? Because it's just above the monolayer formation of water molecules on the surface of Gobi dust. That was evidenced in former studies. I think we presented in dust. The threshold is 15%, so we are just above to make sure to disturb the uptake of acetic acid. So these results are co-adsorption, water plus simultaneously acetic acid. We can look for the adsorption isotherm of the reversible fraction, dry for black and colored for 20% RH, and the irreversible fraction, the same color code. So what we can see is that the presence of water in the stream promotes the release of acetic acid from the reversible fraction. And the presence of moisture also hinders the uptake of the irreversible fraction of acetic acid. So looking at this, we can say, well, it's the classical competitive adsorption. Maybe water blocks the active site and then it prevents the adsorption of the irreversible fraction or promotes the desorption. We can be a bit, we can be further in this way and say maybe in an atmospheric scenario, the dust can be covered by acetic acid first and then moisturize air mass. So what would happen? To simulate this scenario, we have taken up acetic acid under dry condition on the Gobi dust. So it means that we have completed 100% of what could be adsorbed irreversibly under these conditions. Right. In parallel, we can have a look to the drift spectrum of acetic acid adsorbed on the Gobi dust, dry air. If now we shut down acetic acid, I mean the air mass of acetic acid are over, and we enter a moisturized zone. So we can flow 20% RH. What we can see is that the irreversible Takenov fraction get finally dissolved in the presence of 20% RH. Okay, and it's 67% of that supposedly irreversible fraction that get dissolved by the arrival of water molecules, and only 33% remains. We can do an equivalent experiment, but with only 0.8% RH in the drift cell, because it would not sustain 20% RH, but we can already understand what happens. In fact, what we can see is that indeed, when water is sent into the reactor, most of the peaks corresponding to molecularly adsorbed species are decreased. Only one peak is not decreased. This is the one that corresponds to acetate, which is not disturbed at all by the arrival of water molecule on the surface. So clearly these acetates are not impacted by the fluctuation of environmental conditions. So to wrap up on these results, how does acetic acid can affect the surface of Gobi dust? First, what we have done is that we have identified the different natures of the species present on the surface of Gobi dust. We have molecularly adsorbed acetic acid, but we also have chemisorption and the formation of surface acetate in a binded mode. Flushing with zero air removes most of the molecularly adsorbed acetic acid, but it does not affect acetates. And if water molecule competes with molecularly adsorbed acetic acid on Gobi, it shows no impact on the acetate while increasing the moisture content. So typically we can consider that we can get quite easily, even for the lowest concentration of acetic acid, something like 5×10 to the power 13 molecule per centimeter square of acetates that can be stored on the Gobi dust surface. And this questions further behavior of the dust, because it can happen for acetic acid, but it may be also the case for other organics. And we can further question how it would impact further uptakes of other gases, not only organics, but maybe ozone or else, or how it may impact also hygroscopic properties. And I thank you for your attention. I've got in the chat. I will discover the question myself. Is it the question of Jean Bouchy? No, maybe another one. TO2 seems extremely high. Is this representative of Gobi dust? Yes, I mean it is representative of Gobi dust in that they are natural samples that we have characterized with different fractions. Did you do XRF? Yes, we can provide the content of titanium inside this. I agree with you on the fact that the phase assigned to where titanium is supposed to be involved is titanium dioxide, but titanium dioxide is just a formula. It does not tell us about the allotropic phase. So it should be interesting to go further and to check whether it's really anatase or rutile or brucite or any other phase. So this is typical of Gobi dust. This is something that we have already seen, and I don't want to spoil the talk of Jérôme, but we can reach even higher content of titanium element and titanium oxide phases in volcanic dusts. So if you're already surprised by the load of titanium in Gobi dust, you will be even more surprised in volcanic dusts.
Can I complete some?
Yes.
We got some results about the XPS of this Gobi dust actually. So what Frédéric was mentioning with these phases were in the bulk. Now we know these are very recent analysis. Actually, we have that the content of titanium on the surface is significantly lower. So we are at around, let's say, below 1% on the surface, but in the bulk is.
This is another very, very important aspect, the way it distributes inside the particle. Is it in the bulk, or is it really accessible in the surface? That's true. Any other question? I don't see any question online. Is there a question? Yes, Michel.
Thank you for this quite interesting talk, very detailed and careful investigation. My question is, can you tell me which species is absorbing? Is it acetic acid monomer or is it a dimer? If it's the monomer, what is the role of the Lamia-insured mechanism? Roaming on the surface, finding another one to make the dimer in situ.
You're talking about the way the dimer is produced. We don't really know. What we know is that it's a sequential process. We need to go for the monomer first to get the dimer thereafter. So if it's a Langmuir-Sherwood process, it means that both species leading to the dimer needs to be adsorbed first. So it could be conditioned by the fact that two monomers are in a close vicinity, and then with a sufficient surface coverage, it may trigger the formation of dimer. I'm not sure about that. It sounds good to be a Langmuir-Sherwood mechanism, but we cannot rule out that it could be allyridial also mechanism. It could be just monomer adsorbed on the goby dust and acetic acid coming from the gas phase directly interacting with the already adsorbed acetic acid. So far, I don't know. I would go for Langmuir-Sherwood because it's yes, that's what happens for most of the time. And one way to determine whether it would be Langmuir-Sherwood or not is that sometimes this kind of mechanism are triggered or initiated with a threshold surface coverage. It means you have to be populated enough so you can have several dimer in a close vicinity to, okay, now we can go to the dimer because we're saving space. So it could be an experimental way of determining this. Concentration dependence with the gas phase, but not only surface coverage dependence, populating the surface of goby dust with various amounts of monomer adsorbed and seeing when do we start getting the dimer. That's the kind of process that we have already seen, for example, during NO2 absorption on titanium dioxide. It's triggered. The formation, the disproportionation toward NO and NO3 is a condition to threshold surface coverage.
Is there any other question? I just have a comment, Frédéric. It will be very interesting to study an unpolar molecule and the impact of water on the desorption. This goes also to Michel about the recent experiments with lovely limonene. This is something that we'll discuss later.
If it goes with limonene, you know I'm okay with it.
Okay, now we can move to the next presentation. It's going to be online.
Oh, yes.
Yes, it will be, well, it's recorded by Ines Tomašek, and the title of the presentation is Interaction of Volcanic Ash with Anthropogenic Pollution, Screening for Presence of Deleterious Organic Compounds on Ash using the Calut bioassay. It's going to be live. Hi, Ines.
Yes. Hello, everyone. I hope you can hear me okay and see me. I will try to share my screen.
Yeah, go ahead.
Okay, so can you see my screen?
Yeah, yes, very, very clear. You can share, you can make it in presentation mode. Okay, perfect.
Yes, okay, perfect. So thanks, Manolis, thanks, Frédéric, thanks, Saverio, honestly, for making this exception for me to do this presentation online. I would just quickly introduce myself, and then I will explain you why this is an exception. So I'm a postdoctoral research fellow presently at University of Clermont-Ferrand, and I will present you the work that I did while I was still in Brussels at the Vrije Universiteit in Brussels, and this was a collaboration also with Manolis. So just to kind of excite you about this talk, and you can clearly already see that I will talk about volcanoes. Why I am late with my presentation and doing this live is because I spent last 5-6 days at La Palma, where there is an ongoing volcanic eruption. It was really super exciting, super crazy. And this is just to show you the diversity of a phenomenon that is volcanic eruption. So what you care about right now, most of you, I guess, is the production of what you call dust. what for me are so volcanic material and volcanic ash. And so basically what you can see on this picture is the lava fountaining event, which is kind of ejecting rather coarse particles, like centimeter size and bigger, sometimes even bombs. And basically this material is then flowing down the slope of the mountain, basically creating lava flows. And this lava flow actually reached the west coast of the island, creating a delta and basically expanding the island from what I now heard by 10 hectares actually. And so also what happens at the entrance of volcanic, well, of volcanic material, of this hot material into the sea is creation of a lot of basically steam, but also some potentially toxic gases. So this is just to show you how diverse atmospheric species are created during volcanic eruptions and associated events. So what I'm particularly interested in and why I went there is the volcanic ash, basically production, and volcanic ash fall. So basically we could kind of see this is taken from a terrace of our house where we were staying. And basically the ejected material, so ash, was being dispersed over settlements, basically, where people live. And so one of the major aspects of my research is actually the health aspect. So how a volcanic ash can actually impact human health. And so I was there to sample ash at different locations. And then maybe at the next conference, I will share the results of this research. But now to get back to some general definitions. So to actually say that volcanic ash, so it is a type of mineral dust because it's a mixture of fine particles. So for me, when what we defined as volcanic ash is particles less than 2 millimeters, and they are basically composed of fragmented volcanic rocks, minerals, and volcanic glass. So as you can imagine, and as I just showed you on all these pictures, is that volcanic emissions are sources of natural air pollutants, including diverse range of gases, with one of the principal gases being sulfur dioxide, and fine particulate matter. So some eruptions, Plinian style eruptions, such as this one with the big column of ash that is being dispersed, they can contain quite a high amount of, sorry, there was an echo. I suddenly have an echo. Sorry.
We can hear you, Ines. We can hear you.
Yeah, but I can hear myself very loudly. I hope it's okay. Okay, it's better. So yeah, we have a particulate matter less than 10 microns in diameter and less than 2.5. So these are your classical air quality monitoring particle sizes. Why? Because they can have quite some implications towards human health. So basically eruptions contribute to poor air quality, specifically in populated areas, but also in distal areas. Maybe some of you recall the eruption, now it's been more than a decade, but Eyjafjallajökull eruption in Iceland basically impacted whole Europe, disrupted the air traffic, but also monitoring stations across Europe detected PM10 material coming from this volcano. So to make a point is that not only people living near volcanoes are worried about this type of dust and the exposure to this dust, but it's also communities that live hundreds of kilometers away from a volcano. And so my particular, again, interest is the respiratory health. So this will be a bit different presentation in a way that I will show you basically a mixture of all these different fields. I like to call myself a medical geologist, so I basically use principles from volcanology, particle toxicology, and environmental geochemistry. And so in order to basically understand how these ash particles can impact our health, so this is the image of a respiratory system, we mainly and firstly care about particle size of these, of volcanic ash. And so there are other properties of any material in general that can actually impact health. So how it will impact upon contact with the like lungs or other body structures. It depends on the size, so how deep in the lungs we can inhale the material. Depends on the particle shape. We all know the concern about, for example, asbestos because of the fiber shape of this material. It depends on the specific surface area available for interaction. It depends on bulk composition of particles, surface composition, and surface reactivity. So these are the main properties of particles. And so what I focused on in the study I will show you is basically surface composition and surface reactivity. Why are we also concerned? And why do we want to look at these properties? It's because volcanic ash is rarely inhaled in isolation. So as I showed you, in like vicinity to volcanoes in urban areas, we will already have been exposed to urban pollution. And so this is just in summary to show you that studies I did during my PhD, that we basically found that co-exposure to urban pollutants, in this case, my proxy for urban pollutant was diesel exhaust particles, but also a complete gasoline exhaust emission, that their co-exposure in an in vitro experiment with lung cells basically showed us that we could have a pro-inflammatory response in these cells, meaning that the co-exposure to these two types of particles could induce some, let's say, negative response in our lung cells. And this kind of initiated further thoughts. So not just, okay, inhaling both particles, but actually the fact that this phenomenon is present in different countries. So these are just some of the examples. This is not far from you, relatively, where you are right now. But basically another question posed was, when this ash travels through the atmosphere, before it's being deposited on the ground and being inhaled, actually, it interacts. Here is the best example of pollution in Mexico City, that these ash particles interact with urban pollution, and actually there are more things happening. So more species can maybe be absorbed on the surface of ash, not only that originate from the column, but also from urban cities. And so this is where I cried for help to Manolis and some of the other experts in this field, so that we together study, at least to make a first step towards study how such organic pollutants could attach to volcanic ash particles. And so basically the aim of this first study was to estimate the presence. So can we actually even see these organic species on volcanic ash samples, and to actually estimate their bioreactivity, because again, I'm interested in the implications towards human health. And so these are the classical urban pollutants that we were interested in. So dioxin, polychlorinated biphenyls, and PAHs. And basically what we did to estimate their presence on the ash was a bioanalytical approach. And so this picture is just showing you kind of a setup, how it looked like, that I had a plate where I would see the cells. And so this assay is a reporter gene cell-based bioassay. So we have cells that are genetically modified to contain a receptor that is reacting with organic compounds. So basically the principle of this method is on the interaction of compounds that can activate this AHR, aryl hydrocarbon receptor. And this receptor in our cells is responsible for mediating the toxic and biological effects of, well, diverse range of organic compounds. And so basically in our assay, we take the ash extracts, we dose them to the cells, and then we are actually able to quantify the response, because based on this biological reaction and induction of this AHR receptor, basically we measure an enzyme that is produced, and the light response actually in this assay. So the amount of this enzyme that we can basically measure is directly proportional to the amount, but also potency of the AHR active compounds in our samples. So it's kind of a screening to tell you if organic compounds are there and whether they are biologically active. And so we analyzed we had 8 samples from 5 different volcanoes around the world. And the results were the following, that when we analyzed crude extracts, meaning the samples that contained all the extracted compounds, so non-cleaned, basically all of the samples showed AHR activity. And this was basically then looked at basically respective to the reference compound, so BAP, for such polycyclic or aromatic hydrocarbon-like compounds. And so we could see that by the fold activity, that all of the samples in one, let's say in different levels, but they all induce this AHR response, meaning that all of the samples actually contain organic compounds. But on the other hand, when we did the analysis with cleaned up extracts, so samples that were specifically processed so that we can measure the dioxins and PCBs from the extracts, we didn't see as much activity. We could basically, PCBs were below detection limit in all samples, but in some of these samples, we could see a low level of dioxin-like compounds. And basically, the conclusion was that the majority of the activity that we could see, considering that the dioxin activity was low, is that it belongs to PAHs and PAH-like compounds. When we did some calculations to actually estimate the bioequivalent concentrations, sorry, there is an echo. So the bioequivalent concentrations that we determined, when we used them in estimation of human exposure to such concentrations, basically we could see that direct exposure to such levels and to the levels of ash that we assume would be inhaled, that they actually pose a low concern for health. But of course, there is a part that we didn't consider, is the long-term exposure. Finally, we also established a fast and cost-effective method as a first screening of the presence of organic compounds, because in principle, we did not directly determine which exact compounds these are, because for this, we would need to use GCMS and more expensive techniques. But this is also a good way to kind of in the future to, with a systematic sampling, to kind of have a fast screening and see whether we need to do further analysis in more detail. Because basically so far, I forgot to mention, and this is, I think, a topic maybe for discussion, when you read the literature about such compounds, organic compounds, you will always see that they're produced by mainly anthropogenic activity, but also natural activities such as forest fires and volcanic eruptions. But in fact, there is not a study, to the best of my knowledge, that demonstrates production of organic compounds such as dioxin by volcanic eruption. As you could see in my first images, during a volcanic eruptions, there is burning of material around, there is burning of vegetation, and there is burning also now that we see in La Palma with this lava flow, burning of houses, of asphalt. So I think these processes are actually producing such compounds. And then what we want to see is whether they can be attached to the volcanic ash. And I think this is a nice area where we could basically all collaborate. So thanks a lot for your understanding and for listening my talk. For any questions, I'm available now or at the indicated email. Thanks.
Thank you, Ines, for this interesting talk. I think there is, Yeah, there is one question. Did you compare your values of oxidative stress to other assays? For example, oxidative potential assay. This is a question from the chat.
Thanks. So basically, this assay was not your classical. Sorry, is this the question for the calyx, or because what I just showed you, this bioassay is not oxidative stress. So this was not really measuring directly mechanisms of toxicity in the lung cells. But my previous research did. So I'm not sure on which part the question refers to.
Now we have, okay, so probably it was not about, yeah, it was a misunderstanding. So
Okay.
your clarification solved.
Yes, so because we did, so in my past studies, we measured, so the markers of basically potential lung injury. So we measure oxidative stress, cytotoxicity, and pro-inflammatory response of the cells upon exposure to ash. But this now was really a bioanalytical method allowing to kind of see the attached species on ash, and they're also biological potency. So the ability to actually react with the biological molecule that is present in our cells.
Okay, are there any questions? Yeah, Michelle? Yeah.
Thank you, Ines. That was an interesting talk. My question is, did I understand you correctly that you found a way to separate the PCDDs and furans from your leachants? If so, what did you do to the sample to eliminate those compounds in the extracts?
Yes. So basically, now it's been a while that I did it, but so it's the pressurized extraction. So basically in a mixture of hexane and acetone, under pressure, we basically get the crude extract. And then basically we can also take this extract and put it through a cleanup column containing like quartz and few layers that are basically attaching all other compounds that are not basically dioxins. And so there are two also separate columns. It's the separate columns that we basically have dioxins in one extract, and then in another one we have PCBs. It is a chemical basically process with all the different dissolvents that we are able to basically first attach it in the portions in the column, and then dissolve it basically and collect it, and then dose it to cells. So actually, so if you can still see my screen, well, it's here as well. So in my publication, we have this procedure explained, but any kind of also studies on urban PM10 that have used calyx have these explained. Sorry for not giving more detail, but it is possible, yes, to extract them separately.
Okay. Thank you, Ines. Enjoy your trip. Collect a lot of ash and bring some for us, please.
Yes.
So
Thanks.
now we can close the first part of the session. Yeah. You want to make an...
Yeah, thank you. This first session is closed. We are 10 minutes late, so we will restart